Bismillahirrahmanirrahim. Assalamu alaikum class. Here is Dr. Misbah Irshad from University of Education, Township Campus, Lahore. Class, today we are going to discuss the pharmacokinetics phase, which is called as Adme. And under this topic, we will discuss one parameter, which is absorption. The key concept underlying this topic, absorption, are what are the pharmacokinetics phase overall, what is significance of Adme, how is absorption of drug is carried out in a body, what are different factors which are affecting absorption, and what is difference between the enteral and parenteral administration, and how they are done. First of all, the introduction about the Adme. Adme is the pharmacokinetics, and it's the study of the different parameters, and these parameters are collectively called as Adme, and it controls the journey of the drug from its point of administration, and this point of administration can be the mouth, it means that the oral, it may be intravenous, intramuscular, or any other source, and therefore drug travels from its point of administration to its point of action, and this whole journey comes under the pharmacokinetics, which is known as ADME. And ADME is an abbreviation in pharmacokinetics and pharmacology, which is which stands for absorption, distribution, metabolism, and excretion. Here is the overall picture of the pharmacokinetics, the principle of ADME, how the drug is taken or the medicine is taken orally, like it's the capsule, which is taken orally, and absorption means how will it get in, how it will be taken into the systemic circulation or the bloodstream, and then as the distribution, it means that where the drug molecule will go, or they will be transported to the any region in, or any region mean any tissue or the cell or the receptors in the body, and how these drugs molecules are metabolized under the term metabolism. It means that how these drugs molecule will be broken down, and this breakdown will be mainly carried out in liver, and lastly how these molecule will get excreted from the physiological system. It means that how does their metabolites or remain will leave the body. And in fact, this pharmacokinetics is, it describes the disposition of pharmaceutical compounds like drugs within the body. Like if we will start from here, this is shown as small intestine, and enterocytes mean these are the internal lining cells which are known, also known as enteron of the small intestine, through which you can say anything which is metabolized in the stomach and small intestine, they are get absorbed into the bloodstream. It means that this region is responsible for the absorption. And next is distribution. And in the distribution, the drug can go in any particular receptor or the region of the body. And after distribution, the drug will be metabolized, like it's shown here, the liver. Through the systemic circulation, the blood will reach in the liver, and how these drug molecules, which are shown like in blues and yellows, they will be converted into their small, you can say, metabolites or the molecules. And lastly, after imparting their effect or the pharmacological activity on the body, how they will get excreted through the body by means of or by the action of a kidney. And here the yellow circles, they show the nanoparticles, and these are the free drugs molecules which were administered in the body. Next is absorption, and absorption is the passage of the drug from its site of administration into the general circulatory system after the enteral administration. It means that mainly absorption is the term which is particularly used for the enteral administration, and the most common enteral route is by the oral administration. It means that orally the drug is taken into the body. This oral administration, which may be in any form, it may be in the form of suspension, tablet, capsule, and this enteral administration involves the esophagus, stomach, small and large intestines. It means that the whole gastrointestinal tract, which is started from the esophagus. No doubt there is also part or the role of the mouth as well. From the esophagus, some, you can say, absorption occurs in small stomach, and laterally larger part in the small intestine. Here is classification of routes of administration, which can be broadly divided into like topical, oral, parenteral, rectal, and inhalation. Topical means the drugs are applied directly or the topically to the skin or the mucous membrane, mainly for local action, like ointments or the creams. And oral means it's used for the systemic. It means that it's not the local one, it's non-local one. Through the systemic or the blood circulation system, the drug will impart its action, and substance is given via the digestive tract, which is also known as GIT. And next is parenteral. A drug which is administered parenterally is one injected via hollow needle into the body at various sites and to varying depth. Varying depth mean maybe to the certain into the certain muscles, which are bigger one, larger one, or maybe very locally, like into finger, where the muscle strength is very weak, or it may be the shoulder where muscles are stronger one. Then is rectal, and these drugs given through the rectum by the suppositories or the enema. And next is inhalation, and the lungs provide an excellent surface for the absorption when the drug is delivered in the gaseous aerosol or the ultra fine solid particles form, like aerosols in and gaseous source, like when inhalers, such type of the drugs are used. Here is difference between these two routes, which is uh, enterol and parenteral. Under enterol, it's derived from the term or the you can say from the intestine, and parenteral par mean beyond. It's mean beyond the intestine, mean other than the intestine or the this route, GIT route, gastrointestinal route. The drug will be administered in the body so that it can be absorbed into the systemic or the bloodstream very efficiently. And this enterol consists of from mouth to rectum, and it's simple and safe, and no sterilization is required in the enterol source, like drugs which may be capsule or the tablet. They can be taken as such with the water or certain liquid, and therefore no sterilization is required, like sterilization is required in case of um, you can say injection. Everything should be free from bacteria or any other microorganism infection, and therefore it's slow because the drug has to pass through the whole digestive tract, and digestive juices also take their part, enzymes, and lastly it may be some sources of irritants or the in case of emergencies. And in case of parenteral, it's mostly given in the cases of emergencies, like when the patient condition is serious, that drug should be directly administered into the bloodstream. Instead, it should be go through the whole, you can say, gastrointestinal tract. Therefore, it's very rapid one, and no FPM is required, and no gastric irritations, and it may be certain case painful, like injections, and invasive means tending to spread very quickly and undesirably or harmfully into the blood. And asepsis mean the absence of bacteria, virus, or other microorganism is required for the administration of the drug through the parenteral source. And therefore the skilled personnels are required for it, and somehow and sometime more adverse events also happened through it. Here are these enteral and parenteral, you can say, ways by which the drugs are administered. Like in enterol, the drug may be taken orally by some sublingual way, mean through lingual, under, like under the tongue. Some drugs are under placed under drugs, like which are used to control the high blood pressure, and in somehow in cases of you can say heart attack, etc. And rectal and not soluble and enteric coated or slow release formulation of drugs. Is enteric coated means the drugs are like some casing or some layer is also present around the drug in order to its release should be made slow. If its release is quick, it means that it will be metabolized first without reaching its target. The first and the foremost point is that the drug must reach at its target, then it should be metabolized. And then is parenteral. It can be taken via IM, IV, IM, intravenous, intramuscular, subcutaneous, and different rates of absorption, therefore, and different plasma peaks are obtained. And note IV infusers. And next is like IV, IM, and SC are shown over here, and for orally like capsule, and sublingual like under tongue. Then is through skin for local or the systemic effect, node patches, and then through lungs, like in case of inhalation, local or the systemic system. It may be locally or through the systemic system, but it's mostly local, that lungs directly involved. And next is through the vaginal, it's also local. And in the eye, like it's also local, pour some uh, eye drops. Next is drugs which are administered orally. They take about 24 hours to pass through the gastrointestinal tract. It means that in 24 hours they will remain in this GIT. Uh, then they will be, you will say, the absorbed, and they will be metabolized or distributed. And individual transit times for the stomach and small intestines are 20 minutes and 6 hours. It means that the drug, after administration through orally or by taking into the mouth, through the mouth, these will serve, or they will pass nearly 20 minutes in stomach and 6 hours in small intestine. It means that small intestine is mainly responsible for the absorption of drugs. And compounds may be absorbed throughout the length of GI tract, but some areas will suit a drug better than other depending on the nature of drug. Nature of drug, how is drug lipophilic, or how much is the drug polar or non-polar, which type of character it will bears. It means polarity is high or low, lipophilicity is high or low, whether it will go towards the fats or the fatty acids. It means towards the cell membrane. Cell membrane mean towards the tissues. And if it's more, you can say hydrophilic, it means that it will go into the circulatory system very easily, because blood consists mainly of much high concentration of water molecules. And the neutral molecules are more readily absorbed through membranes than charged species, because absorption, because these membranes are made up of fatty acids or lipids and phospholipids, cholesterol, and these all molecules are hydrophobic or lipophilic in nature. And therefore these molecules bear more hydrocarbons or more non-polar character. Therefore somehow we can say that the neutral molecules, drug molecules, they can pass through these membranes very easily instead of charged species. Charged species mean the drugs which may be acidic or the basic in character. Here is shown the membrane, cell membrane, and it's the extracellular space outside the cell. And like hydrophobic molecules, like carbon dioxide, oxygen, nitrogen, and some steroids, because hydrophobics mean They are water repellent. Water repellent mean these are non-polar because water is very polar in nature. Therefore, through these cell membranes, which are mainly hydrophobic or you can say lipophilic in character, they can easily pass. They are shown with a solid arrow. There is no, you can say, much high type of hurdle is in their path. Therefore, oxygen, carbon dioxide, nitrogen, or any other drug molecule which is neutral or it's towards the hydrophobic character, it will pass through the membrane easily. Or the small uncharged polar molecule, like water is the small molecule, but it's like uncharged mean it doesn't bear a formal charge like negative one, two, like and so on. But it's still polar to the lone pair of electron. And cholesterol is also a polar molecule due to lone pair of electron on its hydroxyl groups. Urea, nitrogen, lone pair of electron, and ethanol as well. And they can also pass through the cell membranes, but not very efficiently as these neutral molecules, because they somehow they bear some, you can say, polarity. And water is much more polar. Cholesterol is also very polar, and urea, these molecules. These molecule, no doubt, they doesn't cholesterol doesn't bear the partial positive or the negative charge, but overall its molecule is polar due to the presence of lone pair of electrons. And a large uncharged polar molecule like glucose, sucrose, they are no doubt very large molecule. Therefore, there should be some transport system, like there are some carrier proteins which are responsible for the transport of the glucose across the membrane. And therefore, the uncharged, but they are still polar molecule because they bear the uh, aldehyde or ketonic groups in case of carbohydrates. Therefore, they will not pass through the membrane, they will go away. I said there must be some carrier mediated type of transport system should be over here. And like ions, sodium, potassium, these they will not also pass, but certain like ADPs are responsible for their transport. And therefore, you can say these molecule can easily pass through without any transport or the high energy content, uh, you can say, transporters. And in the stomach, there is also ionization of some orally taken drugs. Like ionization of orally administered aspirin is suppressed in the stomach because of presence of acid, which is produced by the parietal cells which are present in the stomach lining. Because already stomach is acidic, and if acidic species poured in the stomach, then it will not be broken down into its respective, you can say, ions. The ions will be the definitely H positive cation and the conjugate base in case of if acid is poured in the stomach. And if stomach is acidic, then there will be very little or no ionization of the acidic drugs. Like here is shown the parietal cell and the stomach lining. In stomach, there is a large amount of, you can say, H positive cations are produced. And carbonic anhydrase is responsible for the keeping this equilibrium or the balance between the H2CO3 and hydro, uh, which is formed from the carbonic acid from the water and the carbon dioxide. Therefore, the balance between these two species like water and carbon dioxide and carbonic acid is well maintained due to the presence of the carbonic acid anhydrase in the body, or the carbonic anhydrate. And here is shown the that this acid will be ionized in this form. It will be left, and when it will leave its negative charge, definitely some negative or the anionic species will also come inside. When it will come inside, and this proton will go inside when this potassium is going to be coming inside of parietal cell, and this is the lumen of the gastric pit. And this balance is maintained by the this definitely it's the proton pump, and this balance is no doubt maintained by the, you can say, the presence of how much H positive will go outside, how many potassium will come inside, and this transport is through the ATP's energy is utilized. And gastrin is the peptide hormone that stimulates the secretion of this gastric acid and ACL by the parietal cells of stomach, and therefore it aids in the gastric motility. And these parietal cells are responsible for pouring lot of water, ACL in the stomach, and this ACL is responsible for keeping very low pH of stomach. Therefore, overall, we can conclude that the ionization of the drugs which are acidic, their ionization is prohibited or suppressed by the stomach acid. Therefore, ionization of acid carrying species in the drug will not be happen in the stomach. Here is overall picture that as a result, this it is absorbed in this uncharged form, like as such aspirin form through the stomach lining and into the bloodstream in a significant amount. And definitely it's uh, by you can say the phenomena which will operate over here is the common nine effect, because already in stomach there are presence of large amount of H positive, and if these H positive are generated from the aspirin, and definitely their concentration will be high, they will join or the react with these conjugate base to regenerate or resynthesize, regain the aspirin. Therefore, aspirin is mostly or the significantly absorbed in the bloodstream as an uncharged species, as the neutral one. Next are the factors affecting absorption. Here are the factors like polarity of the drug, surface area of the region of absorbing tissue. Absorbing tissue means which region is responsible for absorbing the drug molecule. It surface area can be interpreted in terms of area of stomach, like area of intestine, and the time the drug it will spend in contact with that of absorbing region, how much time it will spend, like how much time it will spend in the stomach, and how much time in the small intestine after getting absorbed into the systemic circulation, and the pH of the medium from which the absorption is responsible to be occurred, and the drug's dosage form, in which form the drug is administered, in injection form, in suspension, emulsion, or in the tablet, in syrup, in which form. And what's the drug particle size, whether the drugs are bigger in size or the smaller one. And what's the aqueous solubility of the drug, how much drug molecules are soluble. And again, the solubility, no doubt it depends on the polarity, it depends on the hydrophilicity of the drug. Here is picture which is showing the polarity of the drug, like the hydrophilic drug, the drug molecules which are polar in nature, they are water loving. Therefore, if it is the membrane, then these, if the drug molecule, they are so much in number, like these are here shown as 2, 4, 6, 7, and 8, 8 numbers, and 9 ones go 3. It means that the drug molecule will pass through the membrane very slowly, because they are no doubt hydrophilic or the less polar. And next is the hydrophobic. It means that these drug molecules are like drug molecule are hydrophobic in nature or the water repellent, and these drug molecules are water loving. Water loving mean these are polar one, and these are less polar one or the non-polar one, because I have said that this is the like membrane, and definitely membranes are you can say the lipophilic, or they are made up of fatty acids. They are less polar one. It means that polar species will pass through less quickly, but non-polar has more ability to pass through. Hydrophobic mean that they are the less polar one. When they are the less polar one, it means that yes, they can pass through easily. And if they are very hydrophobic, it means that their polarity is very less. Then it means that only some molecules are outside, most of them are passed through. It means that overall the polarity of the drug is very important, that how much drug is hydrophilic or the water loving or the polar. It means that the polarity of the drug will definitely not allow it to pass through the membrane very easily. But the less polar drugs or the hydrophobic drugs, it's easy for them to pass through or go through the or diffuse through the membranes. And in absorption, there are paracellular and the transcellular diffusions. It means that when absorption of the drug is going to take place, or the is occurring, then molecules of the drug may be diffused through the paracellular or the transcellular diffusion process. It means that the drug that are very polar, they will tend to be absorbed by the paracellular diffusion. And what is the paracellular? If these are the two cells, and here is the connection between these two cells, and it means that here is the intercellular space between these two cells, and through this intercellular space, like from this connection, the drug molecule is going to pass through. When through this connection, or when there is transfer of the substance through the epithelium, which is between these two cells of the membrane, or the through the intercellular space, then this type of diffusion is called as paracellular diffusion. But in case of transcellular diffusion, there is traveling of any substance, or in this case, in case of medicinal chemistry, which we are discussing here, the pharmacokinetics of drug is so the drug molecule are traveling or passing through the cell. It means that here the diffusion is not between these two cells or not through the intercellular space, but diffusion is through the cell. When diffusion is through the cell, it means that these drug molecules are less polar or non-polar in nature because the cell's outer membrane is wholly or the large amount of like lipids, fatty acids, and cholesterol is present in these all molecules are non-polar. Therefore, non-polar or the less polar drug molecules will be diffused through the transcellular diffusion process when they are going through the absorption parameter of ADME or pharmacokinetics. Next is the surface area of absorbing tissue and time. And the degree of absorption can also be related to the surface area of the region of the tissue over which the absorption is occurring, and the time for which the drug spent. It means that how much time the drug will remain in contact with that region, and therefore if the surface area of any region or the part of, you can say, the tissue from or through which the absorption is going to be takes place, then higher surface area mean there should be high degree of absorption of drug molecule. For example, aspirin will be almost fully ionized in the small intestine. Small intestine means that aspirin will find a bigger, you can say, or the larger surface area, and therefore aspirin shouldn't be readily absorbed in this region of GI tract because its ionization is most, you can say, in larger amount it's in small intestine. Therefore the absorption in this region will not be occurred. However, the very large area of the small intestine, like it's the 300 meters square, together with the time spent in it, is the nearly 6 or everything or every, you can say here, the biomolecule or the metabolite, or in this case the drug, will spend nearly 6 hours in the small intestine, and the average area of the small intestine of healthy individual is 300 meters square. Therefore it results in aspirin being absorbed in significant quantities, and otherwise its ionization dissociation was very large or huge, but due to this parameter of the surface area, this aspirin will be absorbed more significantly, or in larger amount or larger concentration it will be absorbed. And the time parameter is also very important. If the drug molecule will spend large time in any tissue, then certainly they will be absorbed through this tissue very efficiently. Here is shown the small intestine overall. These all are folds of small intestine, that in how many folds, in very large number of the folds, these small intestine is present in the abdomen. And here is shown the small area of cross section of small intestine. It consists of villi, mucosa, and some mucosa, and so on. And if this small section is shown over here, as it consists of epithelial cells, and this epithelial cells pertains or contains large number of microvilli, and these are no doubt responsible for increasing the surface area of the small intestine. Therefore, these are responsible for making larger surface area or providing the larger surface area to any species which can be absorbed through the small intestine. Therefore, many drugs molecule, many, you can say, digestive product of food are efficiently absorbed through the small intestine, and therefore they will get into the bloodstream. And definitely when drug is absorbed through the small intestine to the blood, it will definitely be distributed to the respective tissue very efficiently, or it will go towards its target where it will show its uh, pharmacological activity. And fourth one is the pH of the medium. Like throughout the body, the pH vary. Like there is a different pH of esophagus, and next stomach also have the different pH, and small intestine as well. And the pH of the drug and absorbing medium is also very important one. Like the pH scale is shown over here. Red colors represents the low pH or the acidic one, like the green show the uh, towards the neutrality, and towards the you can say these pH towards the little basic one, small intestine. And therefore, whereas basic drugs in intestine depend on their pH, and when drug is coming from the esophagus where pH is towards the neutrality, and it will get entered into the stomach where the pH is very low, like 1 to 2.5, or up to 5 when fed, when there is something in the stomach. Something means some food item, or carbohydrates, proteins, such type of biomolecules are there. And in the proximal small intestine, the pH rises, and in descending colon and distal small intestine, the pH is higher one. And therefore, the pH is an important parameter which is responsible for the drug absorption. It means that the pH of drug and absorbing medium, uh, both are important. What's the pH of drug? It means that what's the acidic or the basic character of drug, and if it's more acidic, then it will get absorbed into the stomach, because the stomach is most acidic, and therefore acidic drugs will be absorbed through the stomach. And the drugs which are basic in character, like codeine, its basic nature drug, it will be most efficiently absorbed through the small intestine, because the pH of small intestine is towards the, you can say, neutrality, and above it's towards the basicity. If drug is acidic, then what will happen? That here is shown the yellow region will show the ionized molecule, and blue region will show or will represent the non-ionized molecule of the drug. Acidic drugs more non-ionized in acidic pH. It means that if pH is 2, like it's of stomach, or overall its pH show the acidity of that tissue or the region, then blue portion is bigger one. It means that drug will be present in the non-ionized form. It means that drug will be going to be absorbed into the bloodstream in the form of non-ionized form, and the ionized portion of the drug at this pH will be very low. At pH 6, This non ionized molecule portion and ionized molecule is nearly equal one. It means that at this pH, half of the molecule are ionized and half of the molecule are non ionized. And at this pH 6 for the acidic drug, you have to tell me that what's the pKa for this drug. pH is different thing, pKa is the different, and you have to tell me the what's the pKa for this drug, where at pH 6 it's half concentration which is administrated is present in the form of ionized form and half is in non-ionized or unionized molecular form. And at pH 8, like basic pH, when pH is basic, then drug molecule will be larger in the ionized form, and so on. At higher pH, the large number of the drug molecule will be present in the ionized form because the drug is in acidic character or the drug bears the acidic functionalities, and therefore when the pH is basic, and definitely there will be formation of large number of the ionized molecule. It means that yes, pH of the tissue and the acidic or the basic character of the drug is very important for its ionization or non-ionization and then respectively its absorption. Next one is the drug dosage form, in which form the drug is administered in the body. Like if drug can be administered in the solid, in semi-solid and liquid and so on in gas phase. And among the solid, it may be unit solid or the bulk dosage form. Like it can be in the tablet form as the unit molecule as the unit solid, or it may consist of the capsule. And the bulk dosage form mean drug can be applied or taken in the form of powder or the dusting powder, and dusting powder is mostly it's used on the skin or on the wounds, for especially for you can say if for for giving the cure or for providing the cure from irritation, or responsible for the absorbing or the absorption of the moisture or the water molecule. And semi-solids mean like creams, paste, gel, or the suppositories. And in liquid form, like monophasic liquids, like which consist of one phase, like syrup solution. Biphasic mean two phases, like emulsions and suspensions. And gases, like through the you can say nose and through the lungs, the drugs can be administered. It may be in the form of inhalers or the aerosols, like when they are used in case asthmatic patients. Therefore, the drug dosage form is very important for its absorption. And the question may be rise over here, what what's in your mind or what you will think, which drug dosage form is more or the most efficient for its absorption, whether solid, semi-solid, liquid, or the gas phase of or the dosage form is responsible or results in more efficient absorption of certain drug. Next is the form of the drug that is absorbed is not necessarily the form that is responsible for its action. It means that the drug, the form in which the drug is absorbed, it is not the same as that what's it's responsible for the its action. Like benzocaine is absorbed as its neutral molecule but acts in its charge form. Like benzocaine, it's absorbed as such through the membrane, but when it's going to impart its action, then there must be protonation over here, and therefore the Absorption and the drug action is these two different things. It may be absorbed in the you can say the uncharged form, and for its action, it uh, there must be some charge, and this charge is definitely in this case is positive charge or the protonation of the amino group. Next is the drug particle size, and definitely our system consists of very small, you can say molecules, very minute cells, and membranes, and therefore the smaller particle size drugs they show better absorption as compared to the drugs which bears the larger particle size, because the molecules have to be binded like this peptide drug conjugate. It should be built up in the body, and this assembly will be shown like this big ball, which consists of number of binding secondary forces, like it may be pi stacking, hydrogen bonding, uh, can be between these two, like drug and the peptide molecules, and hydrophobic interactions, electrostatic forces. It means that multiple type of secondary forces are responsible for holding these two molecules. And laterally, to show its application like anti-tumor, that how these drugs molecule are efficiently absorbed into certain region, or or how tightly they will laterally bind with the receptors to show their effects. And therefore the drug particle size is very important, and in most of the cases the drug particle size should be reduced or the smaller one. And here is the effect of particle size. Some investigation are carried out that the larger particles, their size greater than 7 micron, they are definitely will be lost in the gastrointestinal tract, and the smaller particles, where size is less than 3 micron, they will be lost in the exhaled breath. It means that they will be exhaled out of the body through the, you can say nose or the mouth, and through the overall lungs. And intermediate particle size like 3 to 7 micron, yes, these particle size is very important and very efficient to reach the actual site of action. It means that the drug molecule should be of this small size, like in microns. They shouldn't be so bigger like in centimeters or the millimeters. And definitely, second is the effect of molecular size. The higher molecular size, lower will be the nasal absorption if the drug molecule size is very bigger one. And good systemic bioavailability or absorption can be achieved for the molecule with the molecular weight of up to 1000 daltons when no absorption enhancer is used. It means that without any uh, auxiliary type of compound, these can easily be passed through, and they can be efficiently absorbed in order to impart or to show their pharmacological effect. Next is the aqueous solubility of the drug, and aqueous solubility of drug is very important, and in a sense that solubility of drug both in water and lipid is very important factor in its to show its effectiveness as therapeutic agent and in the design of its dosage form. And for example, the absorption of drugs from the GI tract into the circulatory system by the passive diffusion mean which doesn't need any type of energy, and drug diffuses from the region of higher concentration to the lower one. It depends on the water solubility, how drug molecules are water loving, how drug molecules are efficiently or effectively absorbed into the water molecules. And moreover, the passage of the drug through other membranes will also depend on them having correct balance of the water and lipid solubility. It means that the drug molecule have to pass through the membrane, unless or until they will pass through the membrane, then how they will be available towards their receptor. Therefore, this balance, not the drug should be much more hydrophobic, nor should be the much more hydrophilic. It means that it should bear some characters of polarity, and it should also have some functional group which should be less polar, or their character is towards the lipophilicity. And therefore a drug's distribution through the circulatory system, and therefore to impart its action, it will also depend to some extent on it having a reasonable water solubility. In addition, to be effective, most drugs have to be administered in the dosage form that are water soluble. It means that drugs are administered in the form which is also water soluble. When soluble means that yes, water can easily take through the esophagus, and that uh, take the drug molecule into the intestine and the stomach, and from the stomach and intestine they can be efficiently absorbed through the uh, you can say bloodstream, or overall from the you can say the lipoproteins lining. Next is the importance of water solubility. That what's the importance of water solubility? That is there any importance for the molecule which are more water soluble? Then yes, there is much more importance for the drug which is more water soluble. Although a reasonable degree of water solubility is normally uh, regarded as an essential requirement for the potential drug, it is possible to utilize poor water solubility in drug action and therapy. It means that some drugs which are poor water soluble, yes, they are important for uh, their action to be shown into the stomach. Like it's the parenteral ambonate, which is used to treat the worms like pinworms and hookworms, which are present in the GI tract. And this drug is insoluble in water. Therefore, due to the water solubility, it definitely it will sustain, or it will remain more of the time in the stomach. When it will give more time in the stomach, it means that it will definitely kill the pinworm and the hookworm. And therefore this poor water solubility coupled with the polar nature of the salt means that the drug is poorly absorbed from the gut. When it will be poorly absorbed from the gut, it means that greater part of this dose will retain in the gastrointestinal tract, and therefore its main drug site, or its main site for its action is definitely the stomach. And therefore these drugs which are used to kill the pinworms and hookworms, they are poor water soluble in nature. And the low water solubility of drug can also be used to produce drug depots, and the drugs which are in the chewable dosage form, and the mask bitter tasting drugs. It means that masking the drugs which have the bitter taste, because the taste depends on the substance forming an aqueous phase. And if it's not forming aqueous phase, it means that they are definitely the lower water solubility bearing drugs. And this is the structure of overall parental ambonate drug, that it's used as the counter ion of the drug compound to definitely affect its dissolution rate. And the presence of the multiple oxygens like in this ambonate, it significantly form the hydrogen bondings, and therefore the hydrogen bondings will facilitate the dissolution of the compounds in the water. But overall this combination in this form will definitely will not enter into the, you can say, the bloodstream. Therefore, much of the time it will sustained or retained into the stomach. Next is the reactivity of the water will also affect the stability of a drug in transit. It means that if drug molecules are going to react with the water molecule, like lignocaine is the drug, water causes its hydrolysis. When water causes its hydrolysis, it means that it will break down the drug molecule. Therefore, the drug molecule will be break down from here. Therefore, this amino group will be present as such, like 2,6 dimethyl aniline, and this whole group will be released. It means that if the drugs molecules are going to react with the water, then uh, it's also a very important parameter for the for like in case of lignocaine. And this hydrolysis by water is one of the main routes for the metabolism of drugs containing esters, amides, and other hydrolyzable groups. And the example of anesthesia as the anesthetic lignocaine drug is shown over here. It means that drug molecule should be efficiently absorbed by the motor, and therefore for imparting their metabolic activity they can also be hydrolyzed through the water molecules. Next is if we will incorporate certain solubilizing group in the structure of the drug, you can say the incorporation of the water solubilizing group in the structure of certain drug molecule, then definitely water solubility can be enhanced. And therefore here is the type of the group which can be introduced, and whether the introduction is reversible or the irreversible, and what's the position of incorporation of these groups, and what's the chemical route of introduction. And if the drug's water solubility is too low, it will pass through the GIT tract without a significant amount being absorbed. And therefore, its water solubility should be higher one in order to be absorbed in a larger concentration through the GIT tract. Therefore, these are some measures or some journal, you can say, ways whereby you can enhance the water solubilizing character of certain drug. Here is shown number first the type of the group. It means that if you are going towards the formation of analog of certain drug, then the incorporation of the polar groups into the structure of compound will normally result in the formation of analogs with a better water solubility, because if polar groups are introduced, it means that water solubility will going to be enhanced when you will compare it with its parent or the lead compound from which these analogs are going to be formulated. And polar groups that either ionize or are capable of relatively strong intermolecular forces of attraction with the water will usually result in analog with an increased water solubility. It means that first you have to introduce a polar group, and if this polar group will, you can say, ionize or it in a other way it have the ability to strongly bind or to develop the other such type of intermolecular force of attraction with the water molecule. And definitely this these two polarity and then laterally strong intermolecular force of attraction will result in the enhanced water solubility of the drug molecule. For example, the incorporation of strongly polar alcoholic groups, amines, amide, carboxylic acid, sulfonic acid, phosphorus, oxyacid groups, which form relatively stable hydrates with the water molecules. and which would be expected to result in analogs that are more water soluble than those formed by the introduction of less polar like ether, aldehyde, ketonic functional group. It means that the drug having ether, aldehyde, ketonic functionality, they they could be replaced with the polar groups like alcohol, amino groups, and amide, carboxylic acid, and so on in order to enhance its water solubility, because these all groups are polar in nature. And therefore by introducing polar group, definitely water solubility will be enhanced. When water solubility enhanced, it means that drug will be efficiently or effectively or significantly absorbed through the, you can say, overall GIT tract or GI or the GIT, gastrointestinal tract. Here is the introduction of carboxylic acid group by the alkylation if it's a drug and it's the intermediate drug. If in its structure, if OH group is resulted in such a way that OH group is treated in such a way that there is introduction of carboxylic acid group by the alkylation, like keeping these conditions, then in this way the solubility or the absorption will be enhanced. And dipsone, which is the antibacterial drug, and similarly it's hydrogen can be replaced by this CH2C double bond O and OH group to give its more, you can say, more polarity, or to give its the, you can say, water soluble character. And therefore we can conclude that the introduction of carboxylic acid group by the alkylation definitely enhance the water solubility or the polarity of the group. And therefore this is also an important step in the lead analog synthesis. And next is the introduction of acidic and basic group. It means that you can introduce acidic or basic groups in the drug molecule, and it's particularly useful since these groups can be used to form the salt. If drug is basic one and you introduce the acidic, it means that you are going to form its salt, and therefore which would give a wider range of dosage form for the final product. However, the formation of zwitterine by the introduction of either an acidic group to the structure containing a base or basic group to the structure containing an acidic group can reduce the water solubility. It means that if salt are going to be formed, then it will increase the, you can say, water soluble character. But if the zwitterine are going to be formed, then the water solubility will be reduced. And introduction of weakly polar groups such as carboxylic acid esters, aryl halides, alkyl halides, because these groups are not significantly polar, they are very less polar, and definitely these groups are called the weakly polar groups. And definitely you will not be able to improve the water solubility, and therefore it can result in enhanced lipid solubility. But for absorption and solubilization, you must be it must be kept into mind, or you must memorize it, that there should be balance between the water solubility, or the, you can say, hydrophilic character and the lipophilic characters as well. And lastly, it's the reversible or the irreversible groups can be introduced. Like the type of the group selected also depend on the degree of the permeancy which is required. The group that you are going to introduce in a molecule which are bound directly to the carbon skeleton of the lead by less reactive, like C, carbon carbon bond, carbon oxygen, carbon nitrogen, they are likely to be irreversibly attached to the lead structure. but the groups that are linked to the lead by the steric linkage, amide, phosphate, sulfate, glycosidic, these all type are the multiple groups, multiple bond containing groups, like they bear the double bond character, like steric, amide, uh, like through the carbonyl, nitrogen, phosphate, sulfate, they are more likely to be metabolized from the resulting analog to reform the parent lead as the analog is transferred from its point of administration to its site of action. It means that these groups which are linked to the lead, they definitely reform the parent lead as the analog will be transferred from its point of administration to its point of action. The compound with this type of solubilizing group are acting as the prodrugs, and so their activity is more likely to be the same as that of parent lead compound. It means that by introducing certain reversible and irreversible groups, that the groups which are more tightly bound or the groups which are less tightly bound or weakly bound to the certain lead compound, then you can improve the, you can say, the water soluble or the lipophilic, laterally lipophilic character in order to be absorbed through the membrane. Therefore the solubilizing group will definitely compound with this type of solubilizing group will enhance the activity of this group. It means that in absorption, absorption is not an easier phenomena. It's very complex one, and it depends on number of parameters. And unless or until the drug is efficiently absorbed through, you can say, through the epithelial cells or through the small intestine into the bloodstream, then there should be the effectiveness of the drugs. Otherwise it will be useless to take the dosage form again and again. Student, it's all about the absorption. And for assignment, you have to list five acidic and basic nature of the drugs and their mechanism of absorption in the GIT. And what are, you can make a list, what are the differences between the bioavailability and bioequivalency. It means that these two terms are not the same. I hope you will go through this lecture and will get enough knowledge. And best wishes. Allah Hafiz.